All right. So, on our previous lecture, we talked about senses, general sensory input, how a lot of it's being processed. If this is your first lecture you're watching for this week, I would go back and watch the other one first. It's just going to help you a lot more with this guy. But we are going to leave just that general idea of senses now, and we're going to go into what are known as special senses. Now, if you remember just going back to the last one, these general senses are what is known as visceral sensations. Okay, and they have receptors all over and throughout the body, and they're usually in simple structure. Right? This is your touch. Your body's able to see if it's in pain, if it's hot, if it's cold. Now, special senses, these guys are what you probably think about the most when I say senses: your smell, your vision, your hearing, all those. So as science has progressed and more are being studied when it comes to senses, they've discovered that these guys are actually different than just general senses. Okay, so your smell, your taste, your vision, your hearing, and equilibrium. And what's anatomically distinct from them is that their receptors, instead of just being all over and simple structure, they're actually concentrated in some specific locations, specifically located in the head. Okay, and commonly associated with embedded in the head of the epithelial tissue. Okay, and that's within this just complex sensory organs, just like eyes and ears. All right, so receptors of special senses, they are concentrated, they have more complex structure, and they are usually in epithelial tissue forming these complex sensory structures. Okay, and as with any type of sensory structural, neural pathways are important, right? Especially for these guys. And it actually forms more of this complex idea than what we just saw with plain general senses. Now, one of the major ones you probably think about is smell, right? And so also known as olfaction. All right, so I'll refer to it as smell. Smell is just, it's a chemical sense. All right, when you are smelling, you're breathing in what are known as odorant molecules. Okay, and these guys are what tell you what you're smelling in a sense. All right, they're what help to differentiate. I'm smelling this that's sour. I'm smelling this that smells like me, and so forth, so forth, so forth. And they interact with receptors. Okay, now not only do they interact with receptors, but the process kind of starts before then. All right, and it starts with being like the odorants needing to be detected by dissolving in the mucus. All right, so that's why, and it's dry out in the wintertime, sometimes your smell is a little off. All right, you may not be able to pick up a lot of smells that you would have if it was more of a less dry humidity. Okay, and what's interesting is smell, just like taste that we'll see, is it's got some intense link to the central nervous system. All right, specifically has some strong links to its emotion and memory of other olfactory pathways. All right, so you'll start to see in a lot of these they develop pathways that are separate from others, and that's how we get our smells and our tastes that are different. All right, now while we're talking about smell as its own thing, smell and taste work together. All right, so that is how you get your idea of flavor and the perception of flavor. That's why if you have a stuffy nose, things just may not taste the same. All right, even if you have a dry nose, like we talked prior, it may just not taste the same because they play in on one another. Now, looking at the anatomy of these receptors, as I had said prior, it is in fact within epithelium. Okay, for this instance, olfactory epithelium, which if you could pick up inside your nose and go all the way to the tippy top, that is your olfactory epithelium. All right, so it covers the cribriform plate and the superior nasal concha, which you kind of see here on this depiction. Okay, now within this epithelium, there's other specialty cells. All right, some that do the same jobs that we've seen in previous lecture. All right, we have sensory neurons, which makes sense, right? Those are makes sense. Those are the ones that get the first signal. Okay, supporting epithelial cells, as the name says, they are supporting either the structure or supporting the function. And basal epithelial cells. You can kind of think of basal epithelial cells as structural support, kind of anchoring them down. And alongside with these epitheliums, the specialty cells, we have glands. All right, they're known as olfactory glands, and that's what secretes the mucus. All right, that's what produces that mucus within your nose that therefore goes and helps to dissolve whatever odorants are there and allows us to get our smell going. All right, and this in a way is explaining how you get teary eyes and runny noses when you have an irritant. So if someone is cooking, you have an immense amount of pepper, what happens is your olfactory glands rev up, okay, and they're interacting with a variety of nervous systems interacting. And in a sense, the same pathway becomes where the olfactory glands ramp up. They start to make more mucus. There's your runny nose. And in the process, they interact with parasympathetic fibers, which connect with into the tear ducts activation, which is leading to your runny nose and teary eyes, that garlic smell. Now looking further at those cell types, the olfactory sensory neurons, those are first order neurons. All right, so just like we talked before, those are the guys that get the signal from the receptors. All right, they're the first one to get that message. These neurons are special because they're considered what are known as bipolar neurons. So they have a dendrite, all right, so that ends, it's kind of like a knob shape. All right, and then they have an olfactory cilia, which is the site of transduction, or the transfer of the electrical charge. Okay, so it goes from the dendrite end to the olfactory cilia in order. Okay, and the axons pass this through the cribriform plate to the olfactory bulb. All right, so the olfactory bulb is what's gonna contain the receptor proteins that is gonna bind to the odorants. All right, so once those bind, that's when we can finally get the signal going. All right, but as I said, there's 3 cells. It's not just one doing all the job. The other epithelial cells have their own role. The supporting, as I had said, those are there for structural support. They actually give nourishment to the tissue and help detoxify harmful chemicals, especially those that you may be breathing in. All right, so either allowing this first line of response within your immune system or your detoxifying system, and they kind of do a similar thing with the basal epithelial cells. All right, so they can also help with structural support and detoxifying, but they also interact as stem cells, and they keep replenishing these neurons and these other cells. So they actually replace these olfactory sensory neurons about every 2 months, kind of keeping, the wheel turning, everything going. As over time cells will begin to die, they'll be damaged by whatever odorant you're inhaling. So this is there to kind of keep that all there, so you're not just going around in a couple months losing your cell. So now the physiology of olfaction or smell, it's also known as olfactory transduction, that begins, as I had said, you have your mucus, the mucus comes in, starts to dissolve whatever odorant you just sniffed in, okay, and then in the process, the odorant eventually will bind to the receptor protein that's on olfactory cilia, okay, and once it binds to that, it activates what is known as a G protein. Now, G protein is just a protein that is commonly interacting with a transmembrane receptor. Okay, so think of it kind of like as a shoe hanging off the receptor. Okay, so in the process, odorant comes in, it binds, G protein becomes activated, it now has its own kind of catalyst reaction that it needs to do, and it goes forth and it stimulates what is known as adenylyl cyclase. Okay, and so in the process of that, it actually uses some energy. All right, uses some energy, ATP, forming AMP. So if you remember, ATP is triphosphate, AMP is monophosphate. Okay, so ATP is used, AMP is produced, and AMP actually becomes what is known as the second messenger. Okay, exactly as you think the name sounds. It is going to carry the message in this instance. It's olfactory transduction, and it's the middle guy between the grounds. Okay, so what it does and why it's so important is that this comes together with a couple other compounds or molecules, and it opens what are known as the cation channels. All right, so cation, positive charge. All right, so it opens those cation channels, and in the process of the cation channels opening, it can now have sodium and calcium start to enter into the cytosol of the cells. All right, so this is a form of depolarization. Now, if you remember from the previous lecture, this helps you to start to gain potential. All right, potential that if it hits a threshold, if that gets reached, an action potential occurs. And the action potential occurs, we can finally have, in a sense, a nerve firing. All right, so a nerve sending a message onward until it finally goes through its job and reaches into the central nervous system. Okay, so just by sniffing, you're activating a variety of proteins and using a various of secondary messengers that, in all, it's leading to a depolarization by sodium and calcium. All right, and then if there's enough, we get an action potential. You're smelling constantly. All right, and this is, as you remember, kind of a form of adaptation where you smell some terrible sulfur smell, you actually start to adapt and kind of get less over it. It's because it's impacting which and how far the action potential is. Now, as we had just said, odor discrimination threshold and adaptation. All right, discrimination. You guys have over 10 million olfactory receptors and 400 different receptor types. Okay, so we need different receptor types because we're not just smelling the same odorants. Okay, so the different receptor types can interact with certain odorants, which sends the message to our brain to tell us this is the type of smell I'm smelling. I smell sulfur. I smell salt. This is what helps us do that. And each neuron is expressed on only one receptor type. All right, so it's not there to have several neurons across the same receptor, which is kind of getting like you consider like mixing the signals. And fun fact, you guys sitting here, you can distinguish just over 10,000 odors. I know you're like, that's salty, that's sour, okay, that's meaty. There's actually about 10,000 odors that your nose alone can distinguish against. It's just you don't really have a name for them. You can't put that to mind exactly what that is. And this perception of those odors, the being able to tell what is this and what is that, that's all dependent on whatever pattern the receptor activation takes. Okay, so it's not a single receptor, because now you got to remember this. These receptors are like a forest. Okay, and there's different types of trees within the same forest. Okay, and you can go to each different type of, you can call it a meadow, and in a different pattern. I go to the meadow 1 first, I go to the meadow 2 first. So it's changing this pattern of receptor activation, not just a single receptor, single spot that gives you your ability to discriminate against smells. All right. Now, threshold and adaptation, it's kind of what I had talked about prior. All right, so we actually have a very low threshold. All right, such as we can detect some smells at a very low concentration. And some of them here, like methyl marcipan, is actually added to natural gas, because at extremely low concentrations, our nose, we can still smell it. All right, so it was actually added in a very low concentration because we were so able to detect it even at a very low concentration that it became a safety warning. All right, so our threshold with smelling, it's quite low. All right, that's why some people, intense smells can actually lead to some negative impacts. All right. Now on the flip side, while it leads to some negative impacts, it may be able to basically get over it pretty quick or adapt to the smell you have. Okay, and so every smell we have a really rapid initial adaptation. All right, about 50% of the smell you're getting hit with, within that first second, you've already adapted to it. And usually after about 1 minute, the adaptation is complete. It's near complete. All right, so that's why if you sit into a room, maybe you're sitting near a trash can with a putrid smell, after a couple minutes, you no longer really start to smell it. It's because you've started to adapt to the smell nearby, and it's all done by a variety of things. One, peripheral receptors. Okay, so the receptors that are involved within the activation, they can then impact that transduction signaling that we saw and change the threshold. All right, so not get it to the point where it is an action potential. Okay, and if it's no action potential, the next thing is the central nervous system can't process it because it's not getting that signal sent there. Now, to put the olfactory pathway into more of a, I guess you could say, larger idea of it, okay, so as I had said, you smell. What occurs is mucus starts to dissolve it, and as it starts to dissolve those odorants find receptors within the olfactory epithelium. All right, and those olfactory epitheliums are assisted by two other cells, or three other cells, okay, two epithelial-based cells and one neuron. Now, once that binding occurs, the olfactory sensory neuron starts to get fired, okay, and how that occurs, the transduction, is via signaling that ultimately leads to a depolarization and a strong enough action potential to send that message. And once that message is finally sent up through the cribriform plate, all right, because it's going up, makes more sense, right? Because you're going straight up into the brain. All right, it goes up and through, it follows an olfactory tract and eventually gets into the pathway all the way down into the cerebrum, okay? So that altogether is the olfactory pathway. And as you can see here, it's a quick summary of what we had just said. Those sensory neurons, all right, they go strictly to the nerves in the olfactory one. All right, axons from that pass into that cribriform plate. Synapses are formed within the bulb. And what happens within those synapses, they actually come together with what are known as mitral cells. Okay, so these are your second order neurons. So if you remember, first order sensories, they start the message. Second order neurons, they're kind of like the middleman there. All right, the mailman delivering the package. And eventually they go all the way on up into the cerebrum, and the axons finally hit, our ability for them to process what may being occurred. And so, actually, when it gets to there, some things happen. Okay. Now, as I had mentioned, when you have this response to smell, you get a lot from it. It affects your memories, it affects emotion, it affects the body's ability to determine the threshold, the body's ability to determine what smell it is. And each of that is kind of correlated to a specific region. All right, so the olfactory cortex, that's located within the temporal lobe, that is your conscious awareness of smell. All right, so that cortex is there for you to say, I'm breathing in right now, I smell this, okay, so I am consciously aware of smell. All right, smell is there. The next is the limbic system. All right, the limbic system is there to allow that cortex to give you conscious awareness that the smell is occurring, but allowing you to get any emotional or memory response to the smell. Say, you had a great memory as a child within a field of dandelions. You go and you smell dandelions. Okay, the odorants go through, fire in, get the threshold, go through the neurons, go up the epithelium into the olfactory bulb and all the way up into the brain. You're aware that the smell has hit olfactory cortex, and then you were aware there was a memory or emotional response, limbic system. But what helped you get there, okay, it was the orbitofrontal cortex via the thalamus. Okay, that's how this information gets there. It was your body's ability to identify the odor. All right, that's a dandelion. I can smell it and discriminate. Okay, so I may be smelling all these other flowers, but no, I smell this dandelion. All right, so each portion has its own key role in kind of putting this pathway all together. Now, one of the unique features with this sense is that it is the only system that we've talked about, and we're going to talk about, that reaches the cerebral cortex without synapsing in a thalamus first. Okay, so that's the only sensory system we'll discuss that does that. Okay, so probably the next thing you think of is taste, right? We smell, we taste, we see. So next up, taste. All right, so also known as gustation. This, similar to olfaction, it's actually a chemical sense. Okay, we get our idea of taste and everything from chemicals, and it's much simply done than what smell did. Okay, you've probably heard of we detect five primary tastes: our salty, our sour, our sweet, our bitter, and umami, which is, if you never heard of, it's kind of like that savory, meaty taste. And each one of these comes from some form of a chemical influence. Our salty is sodium ions, our sour is hydrogen ions, sweets are sugars, bitters are actually some toxins. Okay, that's why, a lot of times if something unhealthy for you, say you accidentally absorb or take some medicine that you shouldn't have, it's going to taste bitter to you. Umami, since it's kind of a savory, meaty thing, meaty systems have amino acids. All right, so it's amino acids that stimulate that. And while we talked how taste is done with both the olfactory, okay, it also has some other sensations that can affect it and actually go back to more of our visceral sensations. And it's our tactile and our thermal sensations. Or you ever try to eat something that was way too hot and you didn't let it cool down? Sometimes you lose your taste, right? You've basically almost fried your taste buds that you have lost your ability to taste. Okay, same with tactile sensations, all right? Judging on if your tongue is touching something that is more gritty versus more smooth. All of this combination of what you're smelling, what you're tasting, if it's hot, if it's cold, that is going to lead into the sense of taste ability to identify it, but also to determine, any memory connection or all that fun jazz. And as we talked about before, cold nose, runny nose, you feel like you lose your taste, but in reality, it's not so much in that instance that it is your just taste in general. It's actually your olfaction that's been impacted that takes away your taste. All right, so common misconception that a lot of people say, I got a cold, I can't taste anything. All right, it's actually, I got a cold, I can't smell anything, so now I can't taste. Now, as I mentioned, taste buds. You've probably all know what taste buds are. You've definitely probably have all heard them. You guys right now have about 10,000. All right, they unfortunately do decline with age. You've probably heard your grandparents or even your parents say, I can't just taste the meat like I used to. I can't taste the sourness anymore." You start to realize some older people just throw a lot of seasoning on their food. It's because the taste buds are actually declining, and it's a way for them to be able to actually get that threshold. All right, but taste buds are not just located on your tongue, while a majority of them are there. They're located elsewhere. But those areas, they're associated again with three different types of epithelia cells. Okay, now our taste receptors, they are commonly found on our gustatory epithelia cells. All right, and they have specific microvilli on them. They're referred to, as taste hairs. Such a great name. And they extend through a taste pore. All right, so think of it literally as a little taste hair. All right, you have a pore on these epithelial cells, and you have these microvilli extending through. So it does in fact look like hairs. All right, and this is what helps form a synapse with those first order sensory neurons. So these epithelial cells, they're the first line of taste. Okay, they're there to start the process. Now, unfortunately, they only have about a lifespan of 10 days, and that's where basal epithelial cells come in, and they keep replacing these guys, so you guys aren't just unfortunately losing your taste within a couple weeks. All right. Now, as last one, while you have ones that just do that, we also have supporting epithelial cells, and that's prior. They're there to help with the structural, the functional support, to keep it all together so that the jobs can get done by the others. Now, taste buds, like I had said, all throughout the mouth. Okay, they're housed in the lingual papillae. All right, and this actually is really beneficial because it helps to increase your tongue surface area. And now, papillae within the taste buds, there's 3 different forms. All right, there's the circumvallate papilla. Okay, so these guys are probably the most dominant, I would say. And then you have your fungiform papillae. So as the name sounds, they are unique because they have a mushroom shape to them, and they're scattered all over the tongue. Okay, so not so much as a cluster of one another. And then the last, they are more on the lateral tongue. Okay, so the lateral side of the tongue. And these guys, they actually start to break down early in life. Now, to just kind of give you guys an idea in a picture format of what we're just talking about, I know it's a very lovely photo, but taste buds, like I had said, are all the way throughout. They are clustered in certain regions, papillae, all right, but you can find them all the way through. Like you're commonly associated with that rib structure. However, it's not just bumps all over the place. They do have intense function that are correlating with their structures, and we'll see that in a bit. And within these taste buds, all right, that you can kind of get a better idea right here. The structure is there and is supported by a variety of different epithelial cells. Okay, so like we had talked before, the basal is there to keep the turnover. All right, and then what's more important out of these whole entire cells, these 3 populations, is that taste pore. All right, so put the taste pore to a memory, and the taste pore is literally a pore, okay, and whatever piece or part of the tongue that you're talking about, carrying the taste bud, and it has these little taste hairs that are hanging out, as you can see in the photo. All right, so these guys are there to bring the receptors to the surface to allow whatever chemical to interact with, and then to have the downstream signaling continue through the cells and into the first order taste neurons. Okay, so just a way to put it into picture. Now, as we kind of talked about, the papillae, there's different ones, right? So the filiform papillae, that actually has no taste buds. All right, it's the most numerous. All right, so sections that do not have taste buds. While, like I said, the tongue is majority, and there's other places. Not every part of your tongue and in your mouth needs to have taste buds. Okay, but they still have function. All right, so yeah, they're not receiving whatever chemical you've eaten to tell you what the taste is, but they all have functions. And now with these guys, they help the tongue move the food around, okay, or the oral cavity, or they give you that texture and that friction. They're more there for the touch style things. And since they're touching things, they have tactile receptors, right? So they don't have those taste buds, the help with identifying taste. They have these tactile receptors that are saying, okay, I have the food to my left side of my tongue. I feel it going over my tongue. I'm pushing it to the right. They're there to help things move around the oral cavity. So, knowing all that, and we finally get to it, what is the physiology of taste, or taste transduction? As I said, all chemicals. All right, we don't just slab in some meat and it's the giant chunk of meat that I taste that gives me that flavor. It is the chemicals within inside the food that we eat. Okay, and so those chemicals stimulate the taste receptors. All right, and so instead of odorants, they're called tastants. All right, so tastants are just odorants and taste. All right, and just like deodorants, they have to dissolve. Now, instead of mucus like we had from our olfactory gland that was putting it into our nostril, I'm sure some of you, like myself, are suffering from too much mucus thanks to the cold. The tastants have to be dissolved in saliva. All right, so it's very similar to our smell. Okay, and the taste transduction occurs on Gustaria microvilli. And the mechanism that this occurs, it actually can change by what chemical, okay, or so and forth, you could say taste, may be occurring. All right, so we've already seen a form of depolarization. That was the whole opening of the sodium channels. The sodium and the calcium came in. The action threshold was hit. So that similarly can happen in our taste as well. All right, and now that's seen a lot with our salty sodium enters the cell via ion channels, our sour as the hydrogen ion is entering via ion channels, all leading depolarization. But our sweet, bitter, and umami, they have a little bit of an extra lead to it. All right, these guys, what happens is while this transduction is occurring, those tastants have had come in, they bind to the receptors, but they bind to a specific receptor known as a G protein coupled receptor, and it activates a second messenger. All right, and that second messenger is what goes forth and causes the depolarization. Whereas the first two, it's the stuff themselves, right? The sodium comes in from your eating, that can lead to depolarization. The hydrogen comes in from exactly what you're eating, it can lead to depolarization. Sweet, bitter, and umami have to have a similar mechanism that we saw in smell, because it can't have this direct contact that the other two had. All right, and as with everything, all the depolarization here, it triggers neurotransmitters. All right, and it leads to these action potentials in the first order neurons, which then allows this to continue on to the central nervous system. Taste coding, threshold, and adaptation. All right, so taste coding is exactly what you think it is. Our ability to understand what taste is and give it what the primary taste may be. You've probably a long time ago have seen the map where certain sections of the tongue correlate to a certain type of taste, whether it be sour, sweet, or bitter. But each of these Gustoria epithelial cells, they detect only one primary taste. So this is where you start to get the coding in the map from. Now each taste bud will contain cells for all 5 tastes. Okay, so it's not one taste bud for all 5. Okay, it's each taste bud but with all the cells. All right, and taste perception depends on the patterns of neural activity. Again, it's not single receptors just like smell was. All right, it's all about patterns of the activity and the changes that may occur. All right, so yes, you'll start to see receptors are important. They kind of get that ball rolling, but not so much you can't lie completely on a single receptor to get this whole transduction done. All right, and what's interesting is, yes, a certain pattern of neural activity does it. Say it's on one portion of my tongue. There's a lot of sodium, so I have a salty side of things. But what can happen is there can be different combinations of receptor activation. Maybe the other side of my tongue has sweet. And so what happens is your body and the neural activity processes one side as being salty, the other side as being sweet, so you get your flavor profile of a salty sweet taste of your food. All right, so different combinations of these receptors and the activation of them leads to a variety of combinational neural activity, giving you the idea that you have this different flavor coming at one time. Now, threshold, we have low and high thresholds when it comes to taste, actually. Lowest threshold is bitter. Now, the reason it's so good that we have this is that most toxins are bitter or sour. All right, so our ability to be able to taste, hey, this whatever I ate right here is quite bitter, this is sour, maybe something's wrong with it, allows us as a survival assistance, okay, especially years ago. Whereas on the flip side, we have higher thresholds. All right, so your salty and sweetness. All right, so that's why sometimes your blood sugar is so high, because it actually takes a while for you to taste the sugar, and to an increased manner that you say, hey, wait, there's too much sugar. If you ever tried to put sugar into, say, your coffee, your hot tea, your sweet tea, there's only a fine line that you'll hit, and it'll finally be over, because you've had such a higher tolerance and a threshold that once you hit it, you've hit it. But as with everything, and all of those, and smell and everything, is we have an adaptation method. Okay, now whereas smell was pretty quick to adapt, all right, taste takes a little longer. And it is dependent on exactly what taste you may be having, whether it be salty or sweet. But the adaptation occurs in the same method, taking about 1 to 5 minutes. All right, and this can lead to changes within taste receptor presence. If the olfactory receptor is forming a synapse, all leading to a change in action potential, which can also lead to it not getting to the central nervous system, not getting a response. Now, similar also to what we had with smelling, is that we need saliva, right? We have to dissolve these taste in so we can, taste. And so if you happen to have a dry mouth, your taste is off, right? So if you don't have enough saliva, the adaptation can also be changed one way or another, similar to how we had it with smell. Now, taste pathway, or the gustoria pathway. Our first order neurons, okay, cranial nerves. Now there's 3 guys, they come into play. Facial nerve is about 2/3 of the tongue. The glosophageal nerve is about 1/3 of the tongue, and then we have our throat and epiglottis. Now, how we get to the brain and how this goes all the way through is, we just talked about our taste buds. All right, taste buds get a response by those lovely taste hairs, okay? And if an actionable potential hits, it allows signaling into the first order neurons. Now in this instance, though, we have several first order neurons. As you can see, we have those cranial nerves. So taste buds signal it, they depolarize, it's gone to the cranial nerves. We are now, all right, into our first order neurons. And now what happens is the nucleus is changed within the medulla oblongata, and projections start to occur from the intake of response. Okay, and what occurs is the projections will either go to a lymphatic system or hypothalamus or the thalamus. Okay, now it does a couple things. Now thalamus, it is our taste cortex or insula. All right, that is your conscious perception and discrimination of taste. So similar to our smell, this is our idea, our ability to know, okay, I'm tasting something. I can tell what this taste is. I can determine it from being sweet or bitter. Okay, so that's what allows us to idea that we are tasting and what it could be. So again, starting at the tongue, starting with those taste buds, starting with the activation, action potential is there. Action potential allows the first order neurons to receive the message, in a sense, allows it to send its message all the way through into the medulla oblongata and up into the central nervous system, specifically into the limbic system, the hypothalamus or the thalamus, ultimately leading into our insula, which allows us to perceive that we are eating and that this is the taste. Okay. Okay, so besides hearing and tasting, vision, as if not the first one, is probably definitely one of the top 3 you think of, right? That's allowing you to sit here and see these slides. And to understand the idea of vision, all right, we need to know exactly what wavelengths are and all that fun stuff. So unfortunately for you all, we're going into physics for a hot second. So vision is all about detection of light. And of course, the detection of light allows us to understand movement, color, and depth. All right, and about 50% of sensoreceptors within our body are all in our eyes. So you start to see vision is quite important for us, right? And large regions of that cerebral cortex, so it's not just sensoreceptors, large regions of the cerebral cortex are devoted to us being able to process what we're seeing, so visual processing. And we can actually detect a lot of different types of light. Okay, so obviously we detect visible light. That's 400 to 700 nanometers. Okay, but we can also start to perceive different types of wavelength. Okay, so I won't go into it too much, but as you can see on the bottom right where we have our general visible light strand, we have our ones that are outside. Okay, and it's all dependent when it comes to color by the distance the wavelengths are. Okay, and that's going to change which color it's going to be. So, objects that appear colored to us because of this, they are reflecting wavelengths at a specific distance. Okay, so these guys would be reflecting wavelengths at a specific distance. That's why we get colored. Okay, white. Now, if we have white, it's reflecting all the wavelengths. So we're just getting blasted with all the wavelengths. It's allowing us to see. Black, it is absorbing all the wavelengths. All right, so if you're at night and you're walking and it's blackout, all the wavelengths have been absorbed. You're not able to process any of the wavelengths to get an idea of the visual processing. All right, so this is the changes that occur when we're seeing certain things. Now, if you were looking in your eyes this morning in the mirror before you're getting ready to come to class, you'll see a couple things. You see your eyelids, your eyelashes, and your eyebrows. Now, you guys have probably always thought, those are just there. They don't do anything. In fact, they're actually quite important. All right, they help to protect your eyes. You ever get something in your eyelash? Well, your eyelash is there to actually help it from not getting in your eye. So they're kind of like a net when it comes to some things. They help to reduce the light exposure. Again, eyelashes help with that to kind of break up some of the wavelengths or the nanometers of wavelengths that are getting into your eye. But it also is helped by eyelids. You're staring at a bright light, you're most likely going to close your eye. All right, so the eyelids are there to kind of act as a shield. And it's also there to help spread lubrication. Okay, because if you've ever had contacts on for too long, if you've ever looked at a computer for too long, your eyes start to get dry. It's because they're losing its lubrication. Okay, so that's when the eyelids need to come into play, and they kind of go hand in hand with tarsal glands. So those guys are there, the glands, to actually apply the lubrication, but not just apply the lubrication to the eye. Lubrication is done in a manner to, yes, keep the eye wet, but also to help prevent your eyelids from sticking, right? We want to be able to open our eyes again, right? That would not be a fun instance. And so they're all important. And when things go wrong, bad things happen, such as styes, right? Or blocking of the glands leading to an intense eye dryness and eyelids being stuck, all right? And another thing that you've probably seen is the wetness of your eye, but sometimes it's not just that it's wet. It actually has a thin membrane that lines the eyelids. Okay, it's called a conjunctiva. It's probably conjunctivitis. You've heard of that. When you look at this is what kind of helps with not want to help, but contributes to the bloodshot eyes. All right, because those vessels within the eyes, all right, they dilate. All right, in the process of dilating, more blood is through. All right, so your eyes look swollen, your eyes look red, hence bloodshot eyes. All right, it's because of this mucous membrane lining and the vessels correlated with. Now, alongside those guys, we have what's known as the lacrimal apparatus. All right, again, something, well, I guess this one you don't really see in the morning, but again, it's something you don't really think it has such a purpose. Okay, and it's there to actually make and drain your tears, right? You ever wondered what the hell is making you cry, or what the hell is happening when my eyes are, dry, and next thing I know, I have tears. This apparatus helps to do that. Okay, and it's interesting to think about is some of you probably just think of tears as just some water that's come out, but it's not just water, it's associated with tons of other things, and it's actually antibacterial. Interesting enough, it has enzymes known as lysosomes that actually help with the breakdown of a variety of things, including bacteria cells. So tears actually can act as an antibacterial solution. Now, it's good when we produce them, or I guess if you don't want to cry, it's not good. But what needs to be done is that we need to drain them. We need to get rid of them. Okay, and it's a lot of the times when it drains, it drains into the nasal cavity, as you can see in this image. And when it drains into this nasal cavity, something occurs. You have liquid going into a nasal cavity that already has mucus. Mixes together, comes a little more mucusy, boom, you have a runny nose. That is why you get a runny nose when you're crying. Okay, or if you just have leaky eyes, sometimes associates with a runny nose. Now, these guys are innervated by a facial nerve. Okay, so just like any type of sense, in a sense, this interaction with these cranial nerves is what gives you an idea to yourself that, I am consciously aware that I'm crying. I'm consciously aware of this, and that's why I'm crying. Now, the eyeball is quite an intense organ. Okay, it has a lot that goes into it, of course, because it does so much stuff. Now, some of the first things we're going to touch on are what is known as extraocular muscles. Okay, so these are 6 muscles that help to move your eye. If you're working from the left, you're looking from the right, up and down. These are the 6 muscles that do it, and it's innervated by CN3, CN4, and CN6. Okay, now these muscles have small motor units. And these small motor units are what allows you to have precise eye movements. I see something to the right, it allows me to have this movement straight to the right. I see something slightly to the right, it then allows me to have precise movement to keep my eye slightly to the right by the movement and contraction of the muscles. And this is all coordinated by the brainstem, the cerebellum. It's all coordinating your eye motion. And it actually converges, or has medial movement, of 2 eyeballs. Okay, because you may be thinking that's one portion of the brain that's somehow having to control one side of the eye, when in reality they converge and helps with the movement of 2 eyeballs. Now, outside of the muscles, the eyeball is consistent of 3 layers. The fibrous layer, the vascular layer, and the retina. Fibrous layer is a superficial layer, vascular layer is the middle layer, and the retina is the inner layer. Now, the fibrous layer, this is the superficial layer that consists of 2 things, anterior cornea and a posterior sclera. Right, the cornea is a transparent coat that's over the iris. All right, it's a major refractive surface. All right, so you've seen it when you've kind of looked at your eyes. Okay, it's over the iris. It kind of looks almost like a clear reflective coating. All right, and it actually assists with focusing light onto the retina. All right, so it's kind of like a little funnel to help to capture the light to allow the retina to process an image. This consists of non-keratinized stratified squamous epithelium, as well as some other stuff, including collagen fibers, fibroblasts, simple squamous epithelium, and it's all important when it comes to what the function does. Now, sclera, that's actually the white of the eye. All right, so if you're looking at your eye, obviously we got colors, obviously we got a gaping hole in the black hole, but obviously we also have a white of the eye. Okay, the white portion, obviously. This is a protective feature. All right, and it's actually the area where muscles attach. All right, so that's where those 6 muscles come to attach to allow the movement to occur. Now, the key thing here is that this does not cover the cornea. Makes sense? The white. And it consists of dense collagen fibers and fibroblasts. So some of the key differences there when it comes to cells is cornea's got epithelium and the other does not. All right, solely collagen fibers and fibroblasts. Now, the vena sinus, that is an opening at the junction of both. Okay, I'll show you what that is in a sec. Okay, so fibrous layer. Say if I'm looking at an eyeball, transverse plane. Okay, we have two things. Cornea. All right, protective little reflective area that goes over top of the retina, the pupil. The vena sinus comes on the side, okay, at, I guess you can say, the edge of the corneas, okay, and your white sclera, all right, all the way through, all the way around. Only portion it does not cover is the cornea, okay, and this is the site, as you can see here, as where the muscle is attaching and allowing for the contraction to either move your eye to the left, to the right, to the up, and the down. Okay, so that is the fibrous layer. Now the vascular layer, as you can expect what this layer is going to be for, it consists of three portions. All right, the first is a highly vascular system. All right, it's what gets the nutrients, all the blood that it needs, anything like melanin that it absorbs from stray light. It's there to kind of keep, I guess, the eyeball happy and fed, in a sense. Now, as I said, melanin absorbs stray light. Okay, so that's what causes brown color, and when it absorbs that stray light, it prevents reflection and scattering of it within the eyeball. So that's what's keeping this absorbent of light to not just be this blurry image. All right, it's acting as a way of a fine focus of like a telescope in a sense. Now the ciliary body, it's got a couple other names, but the body produces what is known as aqueous humor. All right, and it also helps to control accommodation for tightening. All right, so this, what you'll see is this kind of comes in key when it comes to your retina opening or closing with light exposure, if it's too much light or it's nighttime. And lastly, of those three is iris. All right, so it's positioned between the cornea and the lens. All right, so if you remember, the cornea was up top, the lens was on the bottom. That last transverse photo, it's smack dab right in the middle, and this is what controls your pupil diameter. All right, as well as it decides your eye color, but again, that is also determined with the impact of melanocytes. Now, as I was saying, the other two, they play a role when it comes to constriction and dilation of pupils. Now, the principal function of the iris, as I mentioned, is to regulate the amount of light entering through the pupil. All right, it uses autonomic reflexes to do that. It's not something that we think, I got to open my eye a little more. I got to close my eye a little more. When bright light stimulates the eye parasympathetic fibers, okay, of the ocular motor specific nerve, it stimulates the sphincter pupillae of the iris to contract, leading to a constriction. All right, so that's where you get the pupil starting to constrict. Okay, now in dim light, the sympathetic neurons stimulate the dilator, okay, of the iris, and that actually does the opposite. Okay, so it makes that instead contract, and the releasing of the others, and this causes an increase in the pupil size or a dilation. Okay, so when you're looking at the vascular layer, as said, iris, right, smack dab in the middle between the cornea as well as the lens. The ciliary body, now that's again two processes. It's there to help with the opening and closing, or the tightening and loosening of the iris to open and close it, whether you're contracting or not. This is just a looking on version to understand what both those are at. And we have our choroid as well. Now the final layer is the retina. It is probably the one you think the most, and it is the most inner layer of the eyeball. Now it itself has two layers, a pigmented and a neural layer. The pigmented layer is there with a sheet of melanin-containing epithelial cells, and its main job is to help to absorb the light. Okay, because that's the whole function of vision, the way we absorb the light. However, the next thing we need if we're absorbing light is to understand we're seeing, right? So that is the neural layer. Now that is a multi-layered outgrowth actually of the brain that processes that visual data before sending any nerve impulses that form the optic nerve. All right, so this is the processing of the visual signal. So pigmented layer, absorbing light, neural layer, processing. Now there is a couple other major landmarks to be aware of, but understanding that one of the main key roles of the retina are these two layers that really bring it home. All right, so as with any of these layers, there was other cells that played a key role. Okay, and interestingly enough, there's three layers of what are known as retinal neurons within the actual retina. And they're all separated by an outer and inner synaptic layers. The three of them are photoreceptor cell layers, bipolar, and ganglion. And now how these all tie in with one another, okay, because right, we need that first neuron, right, is we get light, comes into our eye, the light actually passes through our ganglion and bipolar cell layers. All right, so those guys are the first, and both synaptic layers before it reaches that photoreceptor layer. Okay, and so again, there's other cells that are there to kind of interact and do some things, and that is the bipolar cellular layer has horizontal cells and amacrine cells, and they are there to actually help direct electric currents that are being received as this light is going through. Okay, so they're kind of like the crossing guards of the electric currents to help with visual processing. Okay, but once that light passes through and it completely reaches the photoreceptor layer, that is when the light can finally be absorbed and then can start to become processed into vision. And this is just a final depiction of what you would see, okay, where you would expect it here. All right, you'll start to see that we are spending a lot of time in retina. All right, so that's where all the magic happens. And why all the magic happens here is because they have what are known as photoreceptors. All right, so those are specialized cells in that layer that starts converting light to nerve impulses. All right, this is your rods and your cones. Right, rods are long on cylinder, cones are shorter, tapered. They both do different things, all important for our vision. Okay, rods are what allow us to see our dim light. Right, they don't provide any color vision, only black, white, and some shades of gray. Cones, those are stimulated in bright lights. Okay, so those are the ones that produce our color. So cones, color. All right, that's how I like to remember it. Now, there are different types of cones themselves. All right, within the retina, we have a blue cone. Okay, that's sensitive to blue light. Start to see a trend with the names. Green cones, sensitive to green light, and red cones, which are sensitive to red light. And these, if you ever realize, a lot of colorblind individuals, they correlate with one of these three colors, if not several or all. That is because that's usually what those three are what our cones focus on. Okay, and it's actually the combination of these color processing that allows us to see the other colors in between alongside with the wavelengths. All right, so interior of the eyeball. That is where we have our lens. Okay, it divides, it's kind of like I guess a fence, pick a fence in a sense you can say, because it actually helps to divide what is inside the interior of the eyeball. Okay, specifically into two segments, anterior and posterior. Anterior is the space that's between the cornea and the lens. All right, that's got two chambers. posterior is behind the iris and in front of the zonular fibers and lens. Okay, so two different segments divided by the lens. As I said, anterior is between the cornea and lens, and it's commonly filled with an aqueous humor that's transparently in water with fluid. All right, so it's kind of where you get the watery-lish looking eyes. You got posterior segment. That's actually the larger portion of this whole entire thing, and it is actually filled with this jelly-like substance. All right, so this vitreous humor. All right, so we have aqueous humor in the anterior and vitreous humor in the posterior. Now, those guys do some key things for the eyeball. Aqueous humor, it's there to nourish, keep the cornea and the lens healthy, because if we didn't have that, we were going to have issues with processing light, absorbing light, getting fine-tuned vision. And it's also there to help maintain pressure within the eye. Okay, obviously pressure is extremely important for something that has such high input into our senses and majority of our sensory receptors. This also helps to drain, okay, because we have to keep this constant flow of this nutritious liquid that, obviously over time it's going to become a waste. So the ability to drain is key with this. And if it's not able to drain, if it's blocked, that's when you get into glaucoma. Okay. Whereas on the inside, the vitreous humor, like I said, gel-like, all in the posterior segment, that's there to actually help your retina stay in position or be in position wherever it needs to be. All right. And interestingly enough, if you're all sitting there or whatever, and you've all seen the floaters in your eye, this is coming from the vitreous humors. All right. It's just debris that's in it. It's usually harmless. And so that whole floaters that you may call them are within this portion, all right, the posterior segment. Now, just looking at the interior of the eye, this is what I was meaning. Our anterior portion, right, because the lens is dividing it. All right, anterior portion, and our posterior segment. Okay, on the flip side, and the lens is that dividing kind of picket fence. Okay, more jello-like viscerous in the posterior segment and more aqueous on the anterior. So, eyes are important, right? We all need them. We need to see. Obviously we can live without them. I know it's done all the time, but they are important. And their development is actually quite interesting, and it's really what helps us to get to the point where we can see. And it starts actually pretty early, more earlier than I guess you would think when it comes to fertilization. About 22 days after fertilization, your eyes had started to begin development. All right, and it has all originated from interactions between several things. All right, mesenchyme, surface ectoderm, neuroectoderm of the forebrain. So all this combination of tissues coming together at that 22-day period allowed eye development to begin. And the optic structures, when they started to form, lateral walls of the forebrain started to form those optic grooves. Okay, and those optic grooves are what expanded into optic vesicles. Okay, so optic vesicles then began to grow toward the surface ectoderm and induce formation of lens. All right, so we're starting to get eyes holes, the lens is starting to form, and the optic vesicles later then invaginated to form optic cups, which then remains connected to the brain via optic stalks. All right, so you're starting to see this almost hollow-like ball structure starting to form with its connection to what would future become its central nervous system. So that's not just done there, right? We have to go further on. And what happens next is you've kind of already got this hollowish ball forming or form that's connected, but we need the lens. The lens development is quite key. And what happens is those placoids that originated during this development, they start to invaginate to form little vesicles, because lenses in the eye aren't just this little contact lens cover that you would expect. They're actually this little tiny vesicle structure. Okay, and those vesicles then they eventually develop into the lenses. Okay, and now all this is provided by tons of blood supply. All right, we need blood to go through, whether it's minerals being provided, nutrients, oxygen, and this is actually where the hyaloid arteries play a key role. And those hyaloid arteries, they degenerate after this is done. All right, so lens is developed. Well, let me go back. The shape of the eyeball is come. The connection into the central nervous system is forming, if not formed. The lens begins to develop because of invagination of the lens vesicles. This is all being supplied by these specific arteries. Now, as this is proceeding, some of the arteries go away. All right, but the remaining of those hyaloid arteries, they help to form the central retinal arteries, right? So the arteries that are in your eyes, all right? And now, lens is there, eye shape is there. We got some of the retinal arteries there, but we actually need the retina and the optic nerve, or else you're just building the boat with no bottom. So, a couple things happen. The inner layer of the optic cups becomes the neural layer of the retina. And the outer layer of that optic cup that was formed, that becomes your pigmented layer of the retina. And what occurs is axons from that retinal neurons, they extend from the optic stalk, or where the eyes would be connected within the back, and transform it into the optic nerve. All right, so they're starting to basically, in a sense, form nerve developments throughout the skull, in a sense, to form the optic nerve. Now, myelination of that optic nerve begins before birth, but is completed after birth. That's why a lot of times, babies aren't too great at seeing, or they take a while to open their eyes, may not keep them open for that long. That's why they tell you not to shine bright lights in your baby's eyes or anything like that. It's because it actually takes some time after birth to then develop. So at that point, all right, we went from this optic cup forming, and then the lens forming, separation between the optic cups, and then the optic cups coming and developing the optic nerve by development of both the layers of the retina. But we need some interior and outer eye layers, right, to really bring it together. And so what happens is that anterior optic cup, it helps to form the epithelia that's needed for the iris and the ciliary body and all the radial muscles of the iris. So it actually helps. It's the key portion, right? Because it's all things that really keep the eye healthy and keep the eye doing one of its jobs, which is moving around. And so the surrounding then, mesenchyme, okay, that's all around that optic cup. That helps form the ciliary muscles and the fibers that are there to help suspend the ligaments. All right, so that helps to form the anterior eye structures. Now, the outer eye structures, the mesenchyme helps with that as well. Okay, without the mesenchyme around the optic cup, it actually starts to differentiate the cells into our inner vascular layer and our outer fibrous layers. Okay, even more so, it begins to form the cornea. All right, that from the mesenchyme, but also from surface ectoderm. All right, so we start to see our outer eye layers, some vascular but also fibrous, start to form. Now, eye chambers and vitreous humor. So as we saw before, right, we have anterior and posterior chambers. Both have different type of aqueous or vitreous humor. All right, so the anterior chamber precisely develops between the cornea and the iris, whereas posterior chamber develops between the iris and lens. That is what helps to get the perfect division of the two chambers. Okay. Mesenchyme enters through the fissure, which forms fibers, okay, that later fill with gel. So that helps to create the vitreous humor on the inside of the posterior chamber. Whereas on the flip side, on the eyelids, the the eyelids form a surface ectoderm and mesenchyme, and within that formation, the mesenchyme helps to form the glands needed for keeping the eyes moist, as well as keeping the aqueous humor where it's needed to be. This is just a quick depiction, just to kind of help put that got in your head. Feel free to look at this as well as in your textbook. It's got a pretty good explanation, but give it a crack on there. I won't go to it. Now, physiology of vision. Okay, so we need to be able to see light. We need to be able to understand that we are seeing, and we need to be able to understand exactly what that is. All right, so image formation. There's three key processes, right? Refraction, accommodation, and pupil constriction. Refraction, bending of light, that is mostly done by the cornea. Accommodation is the lens curvature, which is there to help with near vision and impact on far vision, and pupil constriction. All right, so that improves image sharpness. Like I said, it's kind of like a telescope, and you're kind of opening it up and closing it. All right, that's your parasympathetic fibers and sympathetic neurons. Now, images formed on the retina, interestingly enough, are inverted and reversed. Our brain just actually corrects them. All right, so they don't just come in exactly as we see it. And if not, we can have some errors, or we can have some issues. And then a lot of these refractive errors, so forth, are called, aren't just from understanding image formation. It's actually from mishaps in the development, such as we have nearsightedness, farsightedness, uneven curvature of their eyes, such as astigmatism. All of this stuff, luckily for us nowadays, is pretty corrected with glasses and contacts. Again, a lot of the changes actually do come from the development more so than something that's happened after. So looking at the physiology of light, this is exactly what I mean. Okay, so if you are viewing a distant object, say here in the center, distant object is being viewed, it projects light rays. Okay, now when the light rays will hit the eye, all right, the muscle becomes relaxed, the fibers become tight, and in the process it kind of acts like a mirror where it bends the light itself into the back of the eyes. Okay, and now this type of viewing can change by if it's distant or if it's closer, such as, and you see here, close object viewing. It changes the light rays from the distance of the object. However, the eye itself has methods of adjustment, if not damaged by development or other disease, to accommodate for the changes and to make it so that the absorbing of the light and the sharpness is identical to something that was far away or something that's close. Okay, so the eye is designed to have this unaware response to kind of correct as needed. For this, you guys can look at this. This is mostly just a way to show how actual correction of the eye may occur, or how possible issues such as astigmatism, farsightedness, nearsightedness may impact the vision line. All right, so photoreceptors. Each photoreceptor has three main regions: an outer segment, inner segment, and a synaptic terminal. Now the outer segment, that's where light transduction occurs. And it contains photopigments that are embedded in membranes. This is your rods and your cones. Okay. And the outer segments are continually renewed. All right, so they are constantly being turned over. And then it's actually kind of like a bunch of, the structure looks kind of like if you put a bunch of frisbees and you stack them on top of one another. And when the turnover occurs, what happens is a new one, all right, is put at the base, and the old one that's up top is shedded. So it's kind of like recycling as it's continually being renewing the rods and the cones. All right, now the inner segment it contains the nucleus, the mitochondria, the Golgi apparatus. This is where the metabolic activity is highly increased, and it's all done in a manner to store energy, to get energy, to actually help support the phototransduction that's going to occur. Now, the synaptic terminal, as we'd expect, it's there to help release neurotransmitters, and a lot of times this is glutamate, but it's also there to lead the communication. All right, communication with a bipolar and horizontal cell to allow for the absorption and response of light for vision. Now, this is a quick just kind of idea of what occurs with the receptors and the idea of exactly what I mean. Outer segment, as you can see, the disc stacked onto one another. Our inner segment is your mitochondria, your Golgi, your nucleus, and lastly our synaptic. It's down here. Okay, so this all is circled around rods and cones, right? Photopigments are all light sensitive proteins. All right, so remember that photopigments are proteins and they are light sensitive. That's one of the key characteristics about them. All right, and they're in the outer segment membranes. Rods, 1 photopigment, cones, 3 photopigments. That's that blue, green, and red. Whereas rods only has rhodopsin. Okay, now we looked at some of the layers, we looked at the cells associated and stuff like that, but these photopigments as well have some key components to them. One being opsin, right? So that's a protein portion. That determines what the wavelength is of the light absorbed. And then next is the retinol. So that retinol is a light absorbing molecule. All right, it's a form of a vitamin A derivative. All right, so it's similar structure of vitamin A, but it helps to absorb the light. And what occurs is the main difference in opsin that decides what wavelength is what, and if it's going to be a certain color, if it's dark, if it's not, that's all dependent on opsin structure, because opsin can have different structures in addition of certain molecules, compounds, but the difference is what allows you to see color. Okay, that's the main reason why we can see different colors. Now, photopigment cycle. This is the visual pigment cycle. So exactly as it sounds, our cycle of being able to see via our pigments. All right, it has 4 steps. Our isomerization, that's darkness. All right, so the retinol is in a cis-retinol. So that's that cis-retinol chemical structure where it is bent. All right, light is absorbed, and it converts that retinol into a transretinol. So it goes from a bent structure to a straight structure, trans, cis to trans. The structural change initiates phototransduction. Okay, so transductions of light. And following that is bleaching. So transretinol separates from that other opsin, or that other part of it, the opsin, and the photopigment starts to lose its color. So that's where it gets the bleached name from. It's losing its color. And next, conversion occurs, and that is the enzyme retinol isomerase comes in, and it converts that straight transretinol back to a cis-retinol, or back to a bent retinol. And lastly, regeneration, where cis-retinol recombines with opsin again, and the functional photopigment is then restored. So it's a cycle. Goes back in, it's cis-retinol again, photopigment's restored. We can pop right back up to isomerization, go, 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 go. So that's where you get the cycle portion. And this is just a way to kind of put it here with you guys. The isomerization of retinol, you have it in a cis structure here, gets hit by light, it's in a trans structure where it's straight, separates off from opsin. What occurs is the enzyme comes back in, it takes that retinol, bends it again, puts it right back in, and we start the whole thing all over again. Now, phototransduction occurs, but it can occur in darkness and in light. All right, so you are seeing when it's dark, it's just you're not seeing as well. All right, so you still have phototransduction occurring. So unlike most of those sensory receptors, Photoreceptors are depolarized in darkness and hyperpolarized in response to light. Okay, so far we've always seen, depolarization, depolarization, depolarization. Well, now it's flip-flop and dependent on exactly what response is occurring. And it occurs in 3 different steps, okay, for darkness. A photopigment state, high cyclic GMP levels, and opening of a cyclic GMP gated channel. All right, so photopigment state is just retinal is in the cis retinal form, so that bent retinal form. Photopigments, so rhodopsin, photopsins, and cones are intact, but they're inactive. All right, so they're inactive, there's no activity, and they're located in a disc membrane of the outer segment. All right, so retinal is in cis shape, so bent. All the photopigments are intact and inactive, and they're all located on disc membranes of the outer segment. Okay, so now the high cyclic GMP levels, the enzyme guanylyl cyclase continuously produces cyclic GMP. Okay, and cyclic GMP concentration remains in the outer segment of the cytosol. Okay, and in that instance, it's remaining and staying high. Okay, so the enzyme is just functioning and functioning and functioning and functioning, but it's not being used because it's nighttime, it's an energy intermediate, or for sites, it's darkness. Okay, so because of this, the cytosol remains a high level of cyclic GMP. Now, when we have increased levels of certain molecules, compounds, as you know, you have simple diffusion, facilitated diffusion. It's going to go somewhere normally from high to low concentration, right? So following, we have an opening of gated channels, all right, specifically cyclic GMP gated channels. And what occurs is the cyclic GMP binds to these non-selective cation channels, and as it binds, the channels remain open. And since they're open, it allows cations, in this instance mainly sodium ions, to enter the photoreceptor, and this is where you start to get the depolarization. All right, so next you get what is known as a dark current. So there's just, at this point, a continuous sodium influx. Okay, and that is what is called the dark current. Now, the dark current keeps the photoreceptor depolarized. All right, and because of this, we get what is known as membrane potential. So photoreceptor membrane potential in the darkness is about 40 millivolts. All right, this is significantly more depolarized than any typical neuron at rest. All right, so that's normally about -70. And because of this membrane potential depolarization, the depolarization spreads to the synaptic terminal, and voltage-gated calcium channels remain open. And because of this, calcium influx triggers a continuous release of glutamate, so now we have a neurotransmitter being released. And lastly, this all can lead to an effect on bipolar cells. Okay, many rods, bipolar synapses, glutamate is inhibitory. So if it's producing these inhibitory bipolar cells cannot stimulate the ganglion cells in darkness, which is basically why you don't really see, right? It's not stimulating these cells to continue on. Now, if you look at that in its kind of more picturish format, okay, we have our cis retinal, right? Cis retinal is completely inactive. We're not getting any, light in because it's dark, obviously, so it actually never becomes activated. And in the process, there's no real transduction that is occurring. But a normal enzyme that makes cyclic GMP in the daytime would be making for this process to occur if the light was out. Since the light's not there, it still makes that cyclic GMP, but it's got nowhere to go. So what happens is that cyclic GMP eventually starts to bind to gated channels. Now when the gated channels are bind to cyclic GMP, the sodium comes and it just flows inside. Okay, so this is where you start to get your depolarization. And now that increased large flow of sodium is the dark current, leads to the depolarization, and eventually the depolarization leads to voltage gated calcium channels to remain open, glutamate to be produced and released, but no real, I guess you can say, action potential occurring in the darkness. Now, phototransduction in light is what we expect, right? Vision is light. So when light energy is there, the goal is to convert it into that potential, that threshold within the outer segment of a photoreceptor. So this causes hyperpolarization. Okay, so like I said, this is unique among sensory receptors. Everything beforehand is then depolarization. And it has 8 key steps. And you start to see they kind of feed off one another from dark side of things. It's almost like some of them are flip-flopped. That's retinal isomerization, activation of transducin, activation of cyclic GMP phosphodiesterase, decrease in cyclic GMP, the closure of the gated channels, hyperpolarization, reduced neurotransmitter release, and effect on bipolar and ganglion cells. Okay, so as you saw previously, the whole kind of end up run of this is before darkness didn't excite bipolar cells, and so it didn't inhibit neurotransmitter release. At the end of all these steps, what we'll see is that light actually excites bipolar cells this time, and that inhibits the glutamate or neurotransmitter release. So, first thing, okay, retinal isomerization. Light strikes photoreceptor. Remember, we're in that cis-retinal bent stage. Light hits it. We now go straight retinal, okay, or transretinal, and it activates the photopigment. Photopigments are not activated unless this occurs. Okay, and now the activation of transducin, okay, it's stimulated by this photopigment being activated. And once it's stimulated, it allows a G protein in the disc membrane to become activated and do its job by activation of cyclic GMP phosphodiesterase. Okay, so the G protein interaction and the transduction activation leads to an activation of another enzyme, cyclic GMP phosphodiesterase. Now, this actually reduces cyclic GMP. Okay, because this enzyme PDE, it actually breaks down cyclic GMP. So you see on the previous side of darkness, we actually increase cyclic GMP. So now we're going the other way. We're decreasing it. So as PDE comes in, it breaks it down. The levels of cytosolic cyclic GMP, they fall within the outer segment. Now as they fall, the cyclic GMP gates close. Okay, we saw them open and dark, and now they're closing. Okay, so the reduced cyclic GMP causes cyclic GMP to be removed from the channels, and causing the removal, the door shuts. All right, so those sodium channels are closed. We got no more inflow and no more dark current, or at least a reduced dark current because of the sodium inflow. And at this point, this is when hyperpolarization hits, okay, because we didn't increase sodium levels, we've actually decreased sodium levels. All right, so we're actually hyperpolarizing. So the membrane potential goes from -40 to -65. All right, and this is now hyperpolarizing receptor potential. And now when this goes forth, as I had mentioned, we start to have effects on these bipolar cells, but in a way that we reduce neurotransmitter release, whereas on the flip side, on dark, we increased it. Okay, and it's because hyperpolarization spreads the synaptic terminals, and it actually leads to less voltage-gated calcium channels remaining open. All right, so similar to sodium, calcium influx decreases. Since it has decreased, glutamate release is also decreased. All right, so we've reduced the neurotransmitter release. And further down the road, now we see the bipolar cells being impacted because of this reduction in glutamate, the decrease moves the inhibition of the bipolar cells. The bipolar cells become excited, and since they're excited, they can go do the function. They stimulate ganglion cells. And now those ganglion cells, they are the ones that fire the action potential. I know this is a really long pathway with a lot of steps, but the go-to and what actually it leads to is not just bipolar cells, it is the firing of an action potential from the ganglion cells. And that's how we get into the optic nerve with vision. Again, this is just a way to see it. Light comes in, our cis becomes transducin becomes active. It activates an enzyme that actually leads to breakdown of cyclic GMP. The breakdown of cyclic GMP decreases opening of gated channels, so there's less sodium inflow. The less sodium inflow hyperpolarizes the receptor potential. Okay, and so this leads to the voltage gated channels, calcium channels, to close, and a reduction in neurotransmitter or glutamate release as such. So we kind of talked about this a little earlier, and it kind of goes back to that and how we kind of adjust our eyes, and it's light and dark adaptation. You ever, it's dark, you're walking around your house or your apartment or your dorm room, and then you go into the bathroom and you turn the light on, and you just for a glint just see that your eyes went from pupils really dilated to pupils constrict. Okay, that is what is called as light and dark adaptation. Light adaptation is a rapid decrease in sensitivity. Okay, so instead of increasing, we're rapidly decreasing sensitivity in the light adaptation. And that goes back with what we saw previously about the constriction of the fibers within the retina, causing the constriction of the pupil, leading to a more refined increase of the light. Whereas dark adaptation is a slow increase in sensitivity. That's why if you go into a room and you shut the lights off and it takes you're a little discombobulated to see where you're going, it's because it takes a little time for sensitivity to be obtained, about 30 to 40 minutes. And similar to the pupil restriction, it's the other way around, where the muscles and fibers that are interactive this time open the pupil. Okay. And another way this is done is rods and cones have different regeneration quality, right? Because they need to be regenerated continuously to keep this going. And how this is done and helps actually with light and dark adaptation is the rods, they regenerate pretty slowly. Okay, so in the process of generating pretty slowly, they don't see color. So that's why cones are there for. So rods are there for dark, black, gray, and since they're regenerating so slowly, that's why it takes so long for us to adapt in darkness. We don't have enough rods to kind of come up to be able to handle this adaptation. Whereas cones regenerate quickly, and since they're there to obtain light and see color, which commonly, you see color when light is hitting it, they regenerate so quickly that you're able to recover intensely. Now, retinal processing. Okay, another part of retinal processing is known as convergence. All right, so convergence is interesting in itself, and it's really led by a lot of rods, okay, specifically a lot of them that are within one bipolar cell. And because of this one cell having so many rods within it, it has an intense amount of sensitivity but low acuity. All right, so low able to obtain others, I guess you can say. And it's contributed to what is known as lateral inhibition. We talked about this in passing a little while back in this lecture, but the lateral inhibition or directionality of neural firing, it helps to enhance any contrast when you're looking at something, and light right now like I'm looking at my computer screen. My eyes and the processing of this convergence is enhancing the contrast of the screen. It is also sharpening the edges of the image I am seeing. And if I'm hitting my slides and changing that, that actually helps these macron cells detecting the motion and changing in the light by whether it's yellow writings on the word or black or blue and so forth. So convergence allows us to adapt and improve our sight in a sense by utilizing this immense amount of rods in the cells, as well as this lateral inhibition in the horizontal amacrine cells. Now, a lot of talking, finally got here. The visual pathway. Okay, we've kind of talked about it in chunks. Now, visual pathway, as we've seen, all photoreceptors it starts with. Okay, and interesting, goes to photoreceptors. Then we saw it goes to bipolar cells, and those bipolar cells are then what impacts ganglion cells, and the ganglion cells are what sends that message, all right, that first message. And specifically, ganglion axons form optic nerve, or CN2, okay, and the process has partial crossing at the optic chiasm. I'll show you in a bit what that means. Now, when this is occurring, the optic tracts, which are lateral, okay, to the thalamus, and the optic radiations, which are primary visual cortex, or the occipital lobe, they play into two pieces, I guess you could say, of our recognition and changes of the images. Okay, and more specifically, we have a variety of projections. Okay, so this impacts our eye and head movements. This impacts our reflex and pupil changes, our circadian rhythms. So this process and this projection not only does impact the visual pathway, but plays in the key with other pathways as well. Hence why when your vision is there, you're most commonly able to have a quicker response with your eye and head movements to follow the movement. So this is just showing you kind of an idea of what is occurring. Okay, because remember, everything flops on the opposite side of the body. So if I was seeing it from my left eye, I would have the signal coming in, the photoreceptors hitting, the basal cells, the ganglion cells being activated, hitting up into the optic nerve, into the progression through the visionary midbrain, and all the way on through the optic radiations and into the thalamus. All right, so once we get to the thalamus, the optic radiations, this allows us to process the visual field that is in front of the respective eye. So for instance, left eye in the sense, and the flip side is the right eye, following a similar pathway. Now, the visual cortex is not just the endpoint for this entire pathway. The information gets there. It processes it a little more for us, right? Because if we don't have some editor there to sharpen up the image, it's not going to be as great of a movie to watch. Okay, so our primary visual cortex is there to map the space we see. All right, and it interprets it in a specific way. Okay, but altogether, the way it interprets it, the way it understands it, it's allowing us to consciously see this is a shape, this is a square, this is a color, this is blue, this person's moving, it's motion, it's depth, object recognition. Okay, this is my laptop. It's the visual cortex there that's allowing us to perceive and understand that I can see this is what I'm seeing. So that entire pathway we discussed previously, the goal is to get to this point where the light is absorbed. I can now process what I'm seeing.